Hello everybody and welcome to Cellix's webinar on thrombosis, platelet adhesion and aggregation under shear flow. My name is Dmitry Kashanin, I'm CTO of Cellix Limited, and I'm glad you could join us today. I'm going to start with introduction of Vena8 Fluoroplast biochips and VenaFlux platform, and then proceed to today's application of VenaFlux platform in thrombosis, and in particular whole blood flow assays. And I will give you a quick summary. Cellix's VenaFlux platform allows to mimic venous and arterial flow and in real time visualize cell rolling, cell adhesion and cell migration under continuous shear stress. Similar to intravital microscopy, you can observe cells within the microcapillaries, and the researcher are allowed to perform a functional cell assay where activation of cells can be observed in real time and measured. One of the families of Vena8 biochips, Vena8 Fluoroplast biochip, which has 8 microchannels in dimension of 400 micron wide by 100 micrometer deep, and this biochip is particularly suited for studying platelet adhesion, aggregation and thrombosis. As features of biochips, you can refer to improved optical clarity of this biochip, bright field phase fluorescent microscopy. It can be also used with high magnification objectives, 60x to 100x, and oil immersion objectives. This biochip has very low fluorescent background and plastic matching refractive index of glass. In applications, it can be used for immunostaining of cells, confocal microscopy, or single-cell or platelet analysis. To manipulate liquids to pump cells and whole blood within the microchannels of biochips, we recommend Mirus nanopump, which is ultra-precision pump, covers a range of different shear stresses from very low vascular shear stress to high arterial shear stress. It also can perfuse several channels up to 8 in parallel. It has reversible flow direction and fully computer controlled. During the assay, the images of adherent platelets and aggregated platelets are taken, which can be subsequently analyzed by analysis program. In this case, we recommend Image Pro Analyzer from Media Cybernetics. It can detect aggregates and cells from fluorescent images and count cells, tell information about diameter, form factor, polarization of cells and aggregates. You can also sort the cells and aggregates, and it can provide information on fluorescent intensity of specific aggregate. Cellix's platform allows to observe all 3 major players in thrombosis: interaction with von Willebrand factor, interaction with exposed collagen at the site of injured endothelium, and interaction of fibrinogen receptors. At the site of damaged endothelium, there is collagen being exposed, which allows the binding of von Willebrand factor. Once the von Willebrand factor is bound, the platelets can interact with it via the glycoprotein 1b receptors, and the platelets start to roll on the von Willebrand factor. At the same time, the exposed collagen triggers the interaction with platelet receptor GP1A, GP2A. And also the third factor which influences the platelet aggregation is interaction of fibrinogen receptors, which is GP2B and GP3A, which allows to bind platelets to platelets. For thrombosis experiments, Vena8 Fluoroplast biochips were coated with 10 microliters of specific ligand, collagen, fibrinogen or von Willebrand factor, and then the biochip was stored overnight at 4 degrees. 30 minutes prior to experiment, the channels of biochips were blocked with BSA to avoid non-specific adhesion. To execute flow experiment, the minus pump was filled with physiological buffer, and then connector of the pump was wetted and inserted into biochip. Following this, in the second step, the biochip was washed at low shear stress, and the unbound ligand was therefore removed. After that, in step number 3, the sample was placed into Eppendorf, and the shear flow established by pulling the sample through the biochip into the pump. During the assay, during the flow of whole blood, the several images were taken from different positions of biochip. Typically we would take up to 7 different positions along the channel, and then one video which takes the time lapse of the platelet aggregation from time 0.0 up to 5 minutes. This movie shows the example of aggregation of platelets on fibrillar collagen, and in this case the shear stress was set at 90 dynes per square centimeter, and this experiment would take 2 minutes. As you can see, there is quite a number of big aggregates which consist of several platelets. We analyzed this in Image Pro Analyzer and displayed this data taking into account 2 parameters. First of all, it's surface coverage of all the aggregates, and then the number of aggregates. As you can see in control sample, where the blood was not treated with anything, there's quite a number of big aggregates. So in this case, the surface coverage is quite big, but the number of aggregates is quite small. If one uses the ReaPro inhibitor, which is GP2B3A inhibitor, the picture is completely changed. You can see that there's quite a lot of small aggregates and single platelets, but they cover very small area. So first of all, the area of coverage is reduced, which is shown by this orange graph, but we have increased number of objects. In the case of aspirin, you can see slightly different image, where there's still quite a lot of coverage by the aggregates, but the overall number of objects is increased. And this is also proven by if you look at the mean object area. In the case of non-treated sample, the control sample has a quite big object mean area. In the case of ReaPro, you have only single platelets or several platelets attached together. In the case of aspirin, the number of the area of individual aggregates is reduced. We had also had a look at a comparison of control samples against the ReaPro, aspirin, Canglero, and anti-GP1B antibody, which is all show the familiar effects which is proven in the literature. As for ReaPro, you can only see the single platelets, object size increases, object number is increases, and size is decreasing. In the case of anti-GP1B antibody, you can see both the surface coverage decreasing and object size is decreasing. In the case of aspirin, you have increased object number, but the size is decreased. We also compared different pathways of platelet recruitments to both to von Willebrand factor and collagen, As you can see, we perfuse the whole blood on collagen and on Willebrand factor at 60 dynes per square centimeters. And this is the second one of this experiment, and this is 2-minute time lapse. So as you can see, on the 1 Willebrand factor you mainly have single platelets or small aggregation, and on collagen you have a big aggregates but very small number of these aggregates. Also, we looked at inhibition of platelet adhesion on fibrinogen, and in this case we started with low shear stress at 8 dynes per square centimeter for 2 minutes, and then increased it to 32 dynes per square centimeters and perfused the sample for 4 minutes in total. As you can see, if you don't treat whole blood with RioPro as a control sample, it results in adhesion of platelets at higher shear stress, and if the blood is treated by RioPro 10 microgram per milliliter, you practically have no platelet adhesion from that sample. In summary, Venaflux platform mimics in vivo shear flow microenvironment unlike any other in vitro assay. Researchers are able to assess leukocyte and platelet adhesion and chemokine stimulation and endothelial activation under shear flow. It's robust and easy to use. You can easily coat biochips with a range of different proteins, or also culture cells on the chip. And the result of experiments with Vina8 and Vina8 fluoroplast biochips are reproducible and validated. The system is very flexible. It requires only microliter volumes of whole blood or different cell suspensions to execute the assay, and it can be used with quite a wide range of shear stresses from venous to arterial shear stresses. Thank you for your attention.